The Best of Our Knowledge explores topics on learning, education, and research. Coming up, this week we'll celebrate the best of our knowledge in 2025. We'll learn about a new color that humans normally can't see, a project that created one of the most advanced maps of the brain, and research that uncovers truths to the extinction of the dinosaurs. You're listening to The Best of Our Knowledge. I'm Madeline Reynolds. A new color was discovered in 2025, and it's called Olo. And the few who have been able to see it describe it as a very saturated blue-green. A team of researchers from the University of California, Berkeley, led by Dr. James Fong, used technology called Oz to use lasers to control the photoreceptors in the eye, revealing the new color. In May, Lucas Willard spoke with Hannah Doyle, a research student at Berkeley, who was one of the first to see Olo.
I am one of the few people who's been able to see it. I didn't get to see it for like the first round of subjects that we had, but I have seen it since. And I mean, the experience is you sit into the system. We have to keep our head really still. So you have to bite down on what's called a bite bar, which is kind of like you make a dental impression, and then you bite down onto that. And then you just see like a square kind of like floating in space. Because of like some technical considerations, you actually have to fixate your gaze a little bit away from that square so that the laser light can land on the correct region of your retina. So you're looking away from the square, and then you see a colored square a little bit displaced from where you're looking. And then as for Olo itself, I would describe it as like a very rich blue-green. We do all the visible stimulation with a just regular 543 nanometer green laser. So normally, if you were just looking at that laser, it just looks kind of like emerald green. But then when you turn on this very like specialized stimulation, it's very obvious that the color like changes completely, which is very cool to see.
What was that moment like? And when you saw this Olo square, did it seem unfamiliar to you? Did it seem, I've never seen that color before?
Yeah, so I would say for me, like it wasn't a surprise at that point because we had already been running experiments for a long time, and so I kind of knew
So you're a little
what to
jaded
expect.
about it, I guess. No.
I would say that like because the hue of it is describable, like you aren't it's not an unfamiliar hue, like it's blue-green. The saturation of it is like really what's novel, and one of the ways we're able to like really measure that is we had subjects look at the Olo stimulus and then try to match it with the nearest just like regular monochromatic laser source. And at least the reports that I've gotten from subjects is that the nearest monochromatic light looks almost pale in comparison to the Olo because the saturation level is so different.
There are now products being sold on the internet that I've seen with, Olo paint or Olo this or Olo that. Does any of this stuff that's now floating around on the web, does it even compare to what you saw in the Oz machine?
Yeah, you really can't, reproduce it by nature on like a conventional display or with a paint. So we tried our best to give people a sense of what the hue looks like. That's something you can communicate visually. So it's been really fun to see like all of these kind of offshoots and products and comments that people have given about it.
Now, this also brings to mind the special glasses that look almost like sunglasses that individuals who experience color blindness can wear. And it allows, through filtering the light, people to perceive colors that they normally wouldn't be able to. Is this fundamentally a similar kind of technology?
I would say it's uh what those two things do similarly is that, like, in the Oz system and in those glasses, you can create higher contrast between the signals that the different cone types are receiving than like normally would happen. Um, in Oz, because we can completely like spatially restrict light to the three cone classes, we can achieve more contrast, like in that sense. But there are kind of interesting avenues related to color blindness that we're looking into pursuing with the Oz system.
And that brings me to my next question, which is what are some of the applications of this technology, and what are you in particular interested in exploring here?
Yeah, so one application that I'm not as involved in is, like I just mentioned, looking into color blindness. So in individuals who are colorblind, they're missing one of the three cone types. And one idea we have is to essentially simulate a third cone type in individuals with color blindness using the Oz system. So you could imagine taking someone who's colorblind, putting them in our system, and just pretending that some subset of their cones are now this third cone type that they're missing, and stimulating their retina as if they had that third type, and seeing, can the brain make use of that signal in order to kind of restore like a third dimension to their color vision. But the avenue that I'm interested in, that I've been working on, is using this system to simulate cone loss. So there are some retinal degenerative diseases that cause cone cell death and loss over time in the retina. And I'm interested in how that affects visual function. So in my newer experiments, I'm just taking any subject and I can, in software, just remove like some percentage of their cones. So show them a stimulus, but I'm excluding a subset of their cones. And how does that affect their ability to, for instance, read text or recognize an image, that sort of thing.
Wonderful. And I also heard that you are a master pattern matcher as well in the jigsaw puzzle world. Can you just tell me a little bit before I let you go?
Sure, yeah. So I am a competitive speed jigsaw puzzler. We just had the national championships in early April, and I'm really excited because I came in second. Um, but yeah, most nights I go home and practice some speed puzzling after my research is done.
Well, wonderful. And Hannah, thank you so much for taking the time to speak with me. I really appreciate it.
Yeah, thanks so much.
That was Lucas Willard speaking with Berkeley research student Hannah Doyle in May. Her team's research was published in the journal Nature. You're listening to the best of our knowledge. I'm Madeline Reynolds. The Microns project is a years-long federally funded project that achieved a groundbreaking accomplishment in 2025. Using high-tech mapping tools, scientists were able to reveal the brain's neurocircuitry in never-before-seen 3D images. Artificial intelligence was used to help trace the neural networks of a portion of a mouse's brain. In April, Lucas Willard spoke with Dr. David Markowitz, who coordinated the effort. Markowitz is the former program manager of the International Advanced Research Projects Activity, an organization within the Office of the Director of National Intelligence.
IARPA's original goal in the Microns program was to revolutionize intelligence science. We knew that we would need this enabling data resource. We knew that we would need to make it available to researchers. And I knew when I came to IARPA that I had a fixed window of time to actively support this endeavor, which was 5 years of funding. That's how long ARPA programs typically last. But I also knew as a neuroscientist that this would be a data set that would provide enduring value for driving advances in our understanding of intelligence for a decade, potentially even longer. So we needed to make the data publicly available as soon as possible to as many researchers as possible, and make it as easy to work with the data as possible so that we could maximize our shots on goal for deriving the sorts of insights about computation and intelligence that we wanted to get on a fixed timescale. So I made it my priority from day one of the program that as soon as these data were available, we were going to put them out there so that a high school student in rural Missouri would have the same level of opportunity to make a discovery in these data as a tenured full professor at Harvard University. And so that's what we did about 4 years ago. As soon as the initial revision of the data were available, we put them out there. But your question gets at a really foundational problem in collaborative big science that cuts across disciplines, which is how do you make data not just accessible, but usable, analyzable, without requiring massive amounts of time investment in tool development and skill development. We live in a very special time right now where in the field of machine learning and AI, there's been this, again, I overuse this word, but Cambrian explosion of tools that just lower barriers for people to work with everybody else's data as quickly as possible. And so we drew on some of those best principles from the field of data science and AI. And in fact, the Microns program benefited greatly from work by researchers at Google who developed this visualization tool called NeuroGlancer, which is what enables you and anybody else to go into your web browser and immediately begin visualizing the fine tendril structure of neurons and the long-range connectivity of large-scale circuits in your web browser without knowing anything about neuroscience. And once you've solved that problem, doing deeper level analyses that require a bit more sophistication and scientific training, it's much easier for everybody. So part of my contribution as the program manager at IARPA for the Microns program was we set requirements for data accessibility. We had this back-end data sharing infrastructure, which was called the Neural Data Access System. It defined ways for people to connect your computer with the computers in the cloud to access specific pieces of information about this massive multi-petabyte data set. And that's enabled the development of this entire stack of tools that unlocked these scientific advances that were just reported in Nature.
Now that we have this incredible 3D mapping program that is accessible, it is interactive, and it is available to anybody who wants to click on and use it. Now that we have an actual visualization of neural connections, how can that be applied when doing research on things like Alzheimer's disease?
That is the trillion-dollar question. And I'm not being hyperbolic, because the potential of next generation neuroscience tools and data sets to drive economic impact in AI as well as the biomedical science discipline, it cannot be overstated. And the question that you asked about how do we translate this revolutionary data resource into some meaningful insights that are actionable. People ask that exact same question when the first human genome was sequenced. What do we do with this? We have this amazing thing. Great. What do we do with it? And history tells us that it took decades for people to truly understand how to work with this new type of data. You had to build an entire field of genomic science. And there were many offshoots of that have touched diverse industries from diagnostics to gene editing therapeutics to really sci-fi things like DNA data storage, which is another area in which I ran programs at IARPA. And so for transformational neuroscience data sets like Microns, we are in the process of building new science that will allow us to fulfill that translational vision where we can take insights about computation, insights about the pathophysiology of disease, and translate that into actionable insights for computing, for AI, and for medicine.
So it may be that one day, in addition to right now being able to visualize a part of a brain of a mouse, that we could actually, by just looking at a small part of someone's brain, possibly be able to find the parts of the brain that are in visualization not communicating with each other. Is this possibly a future evolution of this technology?
The short answer is yes. The longer answer is there are so many avenues that are now available to us because of these advances in neuroscience for diagnosing disease. And I'll throw some out there, some of which might make people feel a little bit uncomfortable. So on the more reasonable side is using data from postmortem brains, people who have donated their brains after they pass away, and we know that they have certain diseases. We can use brain mapping tools to identify how the structure of their neural circuits differs from those in healthy controls. And then through non-invasive imaging approaches in patients who are undergoing a diagnostic procedure, you can maybe register what you are seeing non-invasively from a live patient to something that's been mapped in postmortem tissue. And you can say, well, my patient's brain looks more like this early stage Alzheimer's brain, or this early stage Parkinson's brain, or this pre-symptomatic schizophrenia brain. I'm just shooting from the hip here. Another vision of how we might use these tools to aid in diagnostics in the future is actually through brain biopsy. You have 100 million neurons in your brain. You could probably do without about 20,000 of them. And so if you're a person who is experiencing symptoms and your doctor wants to make an informed diagnosis based on more than just a behavioral phenotype, there are tools now for doing very, very small extractions of a very small amount of tissue that you could then put through a neural circuit reconstruction pipeline, including mapping all of the proteins and everything else to unambiguously subtype what type of disease and what subtype this person has. And that may aid in more targeted treatments. And that's a little bit outside a lot of people's comfort zone right now. I'm not sure even I would be comfortable getting a brain biopsy today, but that's the sort of thing at the extreme that these tools could enable.
That was Lucas Willard speaking with Dr. David Markowitz, a former program manager of the International Advanced Research Projects activity, about the Microns project in April. The research was originally published in the journal Nature and Nature Methods. You're listening to The Best of Our Knowledge. I'm Madeline Reynolds. Did you once dream as a child living in a world with dinosaurs? Well, that could have been a possibility if it weren't for an asteroid. This year, scientists determined that fossil evidence from modern day New Mexico disproves theories that dinosaurs were already declining before an extinction event 65 million years ago. I spoke with Andrew Flynn, an assistant professor in the Department of Geological Sciences at New Mexico State University, to learn more.
Yeah, okay. So to like a little bit of background is the end of the very last part of the age of dinosaurs, the Cretaceous period, is something called the Maastrichtian. And so the preceding period is called the Campanian, and the Campanian is really rich dinosaur faunas all over North America. Every area has different dinosaurs. It's sort of a high watermark of dinosaur diversity. Coming into the very end of the age of dinosaurs and the end of the Maastrichtian period, we don't have as good a record. And it's really from, until our research, there was really only one place in West North America with excellent fossils. That's one thing. And you have to have really good age control on your rocks. You have to know how old they are. And so you need both of those things to really understand the patterns. And so what you see in this place in Montana, Wyoming, called the Hell Creek Formation, these are probably dinosaurs that a lot of people are very familiar with. They are very famous. Things like Tyrannosaurus rex, Triceratops, that's where these dinosaurs come from. Jurassic Park highlights a lot of these animals, right? And so it's an amazing place to work, and it's an incredibly rich dinosaur record, but it's one place in the world. And it's hard to draw a lot of conclusions, and so from just one data point. And what it seemed to show in the Hell Creek is maybe there's a little bit of decline in dinosaur diversity going into the end of the age of dinosaurs. And is this a widespread pattern? And the idea was that, well, we don't really find that, we find some of the same dinosaurs in all over Western North America in late Maastrichtian. That's not true, it turns out, is that these rocks in New Mexico, which have been collected for over 100 years, these are not like new dinosaur fossils, but they have new ages on them now. And so previously they were considered to be 70 million years old, which is around 4 million years before the end of the age of dinosaurs, 66 million years ago. And so now we're able to show that they are from this last tiny sliver of time, like this geologic time, this last few 100,000 years before the impact, and they have really different dinosaurs.
So with the research, it states or it kind of shows that dinosaurs from the Southwest and dinosaurs from the Badlands in the North are different. Like the regions, they're just different in some way. So I'd love for you to tell me what those differences are, what the types, were there communities? Think about us today, as people. Was it similar in that way?
Yeah, so it's very similar in that respect. So we call them bioprovinces. Think of them more as associations of animals. And so in the north, you have your Triceratops, your T-Rex, and something called a Montosaurid, which is a big duck-billed dinosaur. That the same day we published in Science, they also published a paper about duck-billed dinosaurs. It's been a nice tie-in. But you have this province in the north, T-Rex, Triceratops, and Montosaurus. It's a coastal plain. It's fairly high elevation or high latitude today. It's fairly high latitude in the Cretaceous. In the south, you have different things. You have this big long-necked sauropod, Alamosaurus. You have crested hadrosaurs or crested duck-billed dinosaurs, which are different. You have different horned dinosaurs. You have similar predators. T-Rex is found in both places. And then there's a whole bunch of other things that are just different between the two. And what's important is that these differences are present earlier in the Cretaceous period during sort of the high water mark of dinosaur diversity. The same provinces are there. They continue all the way up until the end of the age of dinosaurs. And then after the mass extinction, the mammals in the south are very different than the mammals in the north. And so these provinces are there the whole time. So this really leans credence that the diversity of dinosaurs across North America was probably very similar in both periods, or at least they weren't in sort of terminal long-term decline. They weren't doomed going into their extinction. So they are thriving, is what we said in our paper. So and what seems to be really driving this is climate. And so the climate in the south is just warmer. And so certain dinosaurs, especially sauropods, the big long neck guys, they don't tolerate cold weather. Apparently they don't like it cold. And so they only live in places that are warm. And so that gives really good evidence that climate plays a really important role in the distribution of these animals, just like it does today. This is not a unique part of the Cretaceous. Today, climate plays a huge role in where animals and plants are found. And so this is a deep time sort of evidence for climate being a really important factor in sort of where animals and plants live.
So, due to the diversity of dinosaurs from, different regions, this leads to the conclusion that possibly they weren't declining, like you said, that was previously believed that they possibly were like on their last breath or already kind of dwindling. Yeah. So, do you believe that to be true with this new research? And do you think that they were still declining at some rate, or maybe not? I don't know.
Ooh, I personally don't think they were declining. They seem to be doing great. I think what this really highlights the work we just did is now we have 2 data points in Western North America, and that's it. That's like the whole world. That's pretty much the only really good ones that we have. You need both an excellent fossil record and really good ages on the rocks. I think what this highlights is that we just need more data, right? Like, these are not fossils that people didn't know about. We've known about them for a long time. And so highlighting areas that like, hey, we have these fossil assemblages. Now, places like Texas, the Big Bend part of Texas has rocks of similar age, but the age control just isn't as good. And so we don't know, are they the same age? Are they a little bit different? And this is North America, and then highlighting worldwide, right? And so I know there are efforts in Patagonia, in South America, there are efforts in China, actively in Europe, of rocks of similar age. And we're hoping, I think we'll have a much better picture in the next 5 to 10 years of like this really important period in Earth's history. And people are really interested in it in general. Like the public support, public interest in this has been pretty outstanding.
So do you think if it were not for the asteroid, do you think that there's a possibility that they could have lived for, hundreds of thousands or more years, or even, thinking about today, we could have even seen some roaming around? I know that birds are technically considered dinosaurs, right? I'm talking about the T-Rex or those big guys that we know.
Yeah. So absolutely. I mean, you mentioned birds, and in some ways, the age of dinosaurs never really ended because birds are more diverse than any mammal. They're way more diverse than mammals. There are more bird species than there are mammal species, but we talk about the age of mammals because they're just the bigger things on the landscape. Absolutely. They could have gone on to continue to dominate Earth. I mean, dinosaurs are always kind of poked at as sort of a failure, but they're really not. I mean, they dominated terrestrial ecosystems as the big land animals for over 150 million years. Right? That's a really long time, or it's like almost 150 million years. And so more than twice as long as since the extinction. And so they're an incredibly successful group of animals. And I definitely think that without calamities like this, I'm not saying there couldn't have been one later on that knocks them out or something like that, but there's no evidence that they were, in my mind at least, sort of doomed to extinction.
That was Andrew Flynn, an assistant professor in the Department of Geological Sciences at New Mexico State University. The research was originally published in the journal Science. If you'd like to tell the best of our knowledge what you think, send us an email at knowledge@wamc.org. You can listen to The Best of Our Knowledge anytime as a podcast, downloadable wherever you get your podcasts. If you want to catch up on past episodes, to hear more WAMC National Productions programs on the news, academic study, books, women's issues, the media, and the environment, visit WAMCpodcasts.org. This has been The Best of Our Knowledge, episode 1836. The Best of Our Knowledge is a national production of WAMC Northeast Public Radio. The latest on all National Productions programs is available via the Airwaves newsletter and our flagship station's website, WAMC.org. Until next time, I'm Madeline Reynolds.